Good morning. We'll go ahead and get started as people are hopping on. So thanks so much, everybody, for joining today. I'm Satish Gopal. I direct the NCI Center for Global Health. In 2021, we initiated the NCI Global Cancer Research and Control Seminar Series to feature researchers and cancer control experts who are doing impactful and innovative work in global oncology. Today, we're really delighted to welcome Dr. Laura Fejerman, who will talk about her work related to the genetics of breast cancer in women of Latin American heritage. While we would be thrilled to host Dr. Fejerman at the NCI anytime, her talk is perhaps particularly timely today as we are simultaneously celebrating both National Hispanic Heritage Month and also Breast Cancer Awareness Month. Dr. Fejerman is the Placer Breast Cancer Endowed Chair and Professor in the Department of Public Health Sciences School of Medicine at the University of California, Davis. She's the Associate Director for Community Outreach and Engagement, co-director of the Women's Cancer Care and Research Program, abbreviated as WeCARE, and co-director of the Latinos United for Cancer Health Advancement, abbreviated as LUCHA, initiatives at the UC Davis Comprehensive Cancer Center. Dr. Fejerman completed her undergraduate degree in social anthropology at the University of Buenos Aires in Argentina and earned her Ph.D. in biological anthropology and MSc in human biology at the University of Oxford in England. Dr. Fejerman's research focuses on the discovery of genetic and non-genetic factors that contribute to breast cancer risk and prognosis in Hispanic and Latina women. She built the Peruvian Genetics Genomics of Breast Cancer PEG-NBC study, collaborating with the Instituto Nacional de Enfermedades Neoplásicas in Lima, Peru, which includes approximately 2,000 women with breast cancer. She also leads the Latin American Genetics and Genomics of Breast Cancer Consortium, which brings together researchers from the US and Latin American countries to study the genetics of breast cancer in women of Latin American heritage. Dr. Fejerman is passionate about eliminating cancer health disparities and dedicates part of her time to the development and implementation of cancer education tailored to underserved and under-resourced communities in California. Next slide, please. Before handing over to Dr. Fejerman, I'd like to just briefly review a few logistics for this morning's seminar. First, please save your questions for the designated Q&A time immediately following the presentation. You may type your question in the chat box or use the raise hand feature found under reactions. Secondly, please note that today's presentation is being recorded and will be available on our website at cancer.gov/globalhealth. Finally, you can find information about future presentations on our website, again, cancer.gov/globalhealth, and by following us on Twitter at NCI Global Health. And now we'll begin our presentation. Welcome, Dr. Fejerman, and I'm very pleased to hand it over to you now. Thanks so much for joining us this morning.
Thank you so much. I'm very excited sharing my slides. Can you see properly?
Looks good.
Okay, great. So for today, I decided to it's really conceptual, the presentation, as well as showing some data from research, but really I want to introduce and share results from the genome-wide association study in Hispanic Latinas, and then discuss polygenic risk score transferability from original studies to diverse populations. We'll show you results from applying PRS to Hispanic Latinas in the US and Latin American individuals, and then discuss the complexity of investigating differences and disparities by racial, ethnic category, and genetic ancestry. And the whole presentation, the example is always Hispanic Latina in women, because that's the focus of my research. It doesn't mean it's the most relevant compared to other groups. Breast cancer is the most common cancer in women worldwide. And you can see there's differences in age-adjusted incidence globally. My work and the presentation today focuses on US Latinas, Hispanics, as well as woman in Latin America, I've been always very interested in differences by genetic ancestry in different countries and between different groups of Hispanic Latinas. Also in the US, we know there's variation in incidence by racial ethnic category based on census categories. And Hispanic Latinas have the lowest incidence based on data from 2015 to 2019. And the reason for observed differences in incidence that we get to work from and inspire our hypothesis could be actually due to a variety of factors, including, and we need to keep this in mind, the quality of cancer registries, especially when we look globally. Some countries that we had in the global map are basing the incidence rates that we take as part of an entire country. They are based on one or two registries in specific regions of those countries and are not representative of the entire population. There could be also differences in incidence reported based on quality of census data that we use to calculate rates. And that could be even the case in some of the data we see in the US, where certain populations might not have high quality baseline population size based on migration patterns, for example. And then there's social and biological determinants of difference in incidence. And this could go from competing risks in some regions, or some populations have higher prevalence of other diseases, lower life expectancy, and therefore cancer might not be as high in the list of burden compared to other diseases. There's also difference in access to screening and care that can affect, what we see and who do we know about having cancer. And there's risk factor exposure difference, thinking about environmental factors in the broadest possible sense. Could be behavioral factors, it could be nutrition, and then there's the germline genetics and the epigenetics, which is in a way how I see part of the interaction between environmental factors and biology is kind of mediated by these epigenetic effects. So keeping this, I just want you to keep this in mind when we think about everything that I present today. My work started many years ago, looking at the association between genetic ancestry and breast cancer risk in Hispanic Latinas. First, I was working with data from Latinas in the US, in the Bay Area more specifically. And then we were able to replicate and conduct additional studies in women in Latin American countries like Mexico or Peru. But here I'm just illustrating the high level of variation that we can see in different studies, depending where the samples are from, in terms of genetic ancestry distribution. And these are genetic ancestry plots. If you're not familiar with them, each individual is represented by a bar in the vertical bar. So individuals are in the x-axis, and the y-axis represents proportion of each of the 4 ancestries here that are being inferred. And the blue is the Indigenous American ancestry. And so the data is sorted based on proportion of Indigenous American ancestry. And you can see here that in a sample from Kaiser in the Bay Area, these are Hispanic Latinas in the Kaiser database, there's a lot less Indigenous American, on average, Indigenous American ancestry compared to, say, a study that sample individuals in Mexico or Peru, where the Indigenous American proportion is about 80% in this sample on average. Despite this variation, we were able to consistently show that the more indigenous people were in the study, the lower their risk was to develop breast cancer. Eventually, we pulled together multiple studies, again, from the Latinas in the US mostly, but also including women from Latin America, like Colombia or Mexico. And we identify a genetic variant and in the chromosome 6, region 6q25, near the estrogen receptor 1 gene, with a relatively strong association with breast cancer risk, where the rare allele or the least common allele was protective. And here you can see the signal in the middle of the plot coming up. The reason this polymorphism was not discovered before in populations of other ancestries that had conducted genome-wide association studies before was this basically absent in all populations except populations with indigenous American ancestry. And in Latin America, this is data from the 1,000 genomes, publicly available data. You can see among the pointed populations, which are Colombians, Mexicans, Peruvians, and Puerto Ricans of admixed ancestry, the rare G allele is increasing frequency based on what we know about the proportion of indigenous ancestry in those different populations. And as this was very interesting, especially because it was a protective variant, concordant with the observed lower incidence of breast cancer in women from Latin America or of indigenous American ancestry, we encountered this barrier where this was 2014. There was no data that we could find publicly available to follow up the finding. Cell lines didn't represent, this variant was not present in any of the cell lines. Here you can also see germline data available for, say, further replication of fine mapping in different samples was, again, not available. And TCGA, for example, had very few individuals with indigenous American or what they call Native American ancestry. Actually, 2 individuals in the TCGA breast cancer data set had the protective allele, which didn't allow us to investigate much regarding potential function of this polymorphism. So partly inspired by that, I created the Peruvian breast cancer study. I'm not really gonna talk about that study today because I'm doing a more general presentation. But also that lack of data inspired the creation of what we call Latino breast cancer, which is the Latin American genomics of breast cancer study. And it includes individuals all over Latin America and also investigators in the US who've been working on US Hispanic Latinas or even internationally. And I'm happy to say that we are at about 50,000 samples or individuals included in the study. And many of these individuals are being currently genotyped by NCI, which is very exciting. But why do we care about identifying breast cancer associated variants? And why do we care about doing this in all different populations? And one of the reasons is understanding cancer biology. And in that sense, for example, this protective variant that we wouldn't have found in studies including only Europeans or even individuals of Asian ancestry, this protective variant could potentially give us new insights on biology, and so it's really important to identify variation so we can better understand the disease, eventually develop better targeted treatments or even preventative treatments. And the other important aspect is breast cancer risk prediction and for prevention and early detection. And we've been mostly being able to use genetics to predict breast cancer risk based on genetic variants that are low in frequency, so not common in the population, but strong associated effects on risk. So variants on the upper left of the plot. However, in the last 15 years or so, there's been a great increase in our knowledge of higher frequency variants. So variants that could be more useful because they are present in larger proportions of populations, but they have relatively low associated effects. And the way to use these common variants for prediction is by combining them in what we now everyone knows as a polygenic risk score. And basically you add the risk alleles for the different polymorphisms that are common and associated with risk, but you weight the allele number by the size of the effect. And with that, you can create a score that would have a normal distribution in populations. And the idea is that those in the top percentiles of the distribution would be at a increased risk at levels that could be actionable. Same for the lower percentiles in terms of screening recommendations and surveillance. And before I more specifically talk about these different PRS and risk prediction in breast cancer, I just want to remind everyone that we use the area under the curve as a measure of performance of a classifier, different thresholds. And the area under the curve really reflects how well the model classifies people into, in this case, high breast cancer risk or low breast cancer risk. And on the top panel, the one I call A, you can see a typical comparison of distributions. What in this case, it would be cases and controls, and for the classifier. And you can see there's some overlap, but not a lot. So that gives you an area under the curve of about 70% or higher, which is considered as, you know, good. 70% or higher is considered a good classifier. You know, higher than that is very good. In panel B, you can see the ideal perfect classifier where there's no overlap when you compare people with disease, without disease in the distribution of that classifier. And then you have a really bad classifier, which is at basically 50% of the area under the curve, where there's complete overlap. So that classifier doesn't allow you to divide people into groups based on risk. And below, in the unlettered panel, you can see a distribution of polygenic risk score. This is from Nevada 2015, where breast cancer cases are compared to breast cancer controls. You can see it looks kind of not very exciting. This is what we're working with, but that translates to an area under the curve of about 60%, between 60 and 65%. And so that would be in the satisfactory region. It's not ideal, but it's also helpful and can identify people at elevated risk. So this is from the Mavada paper, and I just highlighted at that time they were testing different approaches to select polymorphisms to include in polygenic risk scores, and the level of strictness in how you select the SNPs kind of decreases as you increase the number of polymorphisms. What they show here is that a PRS that included about 300 polymorphisms had an area under the curve of about 63%. If they were more lax in the threshold for what to include in the PRS and included almost 4,000 SNPs, or 3,800 SNPs, the improvement wasn't gigantic, but they got it to, if you round, it would be 64%. And just to really prove that this could be helpful information, even if there's a lot of overlap in the distribution, here we can see the lifetime absolute risk, different ages, and the percentiles of the PRS with different colors. So if you're above the 99th percentile, you get to be at a lifetime absolute risk of about, by age 35, of about 30%. That's similar to some of the intermediate penetrance rare variants that we do use for screening recommendations. And if you take into account the threshold for deciding, when to start mammography, marked by the red line on the right with the 10-year absolute risk, you can see that for some of the percentiles, the lower percentiles, they actually never cross that line. So there could be people in the population that wouldn't benefit from screening because of the risk being low, and some people that might benefit by starting screening earlier. Instead of at 45 or 40, they could start at 35. And there's a current trial testing impact on survival of using polygenic risk score and other factors for risk prediction as a way of stratifying women into groups for screening, trying to see if a more tailored screening recommendation is better or not than the general population recommendation of starting screening between 40 and 50 years of age. So the question I have is, can we use PRS built in populations of mostly European ancestry to apply it to other populations or people with different ancestries? And this is from a paper by Martin et al., where they show, this is for mostly anthropometric traits, and the reference is the European ancestry based PRS. And when they apply the same effect sizes and polymorphisms to predict in other populations, you can see that the prediction accuracy goes down. And the worst case is for populations of African ancestry. So the question is, can we apply the same PRS for breast cancer to Hispanic Latinx individuals in the US and Latin America? And for this work, I'm collaborating with multiple people. Here I'm showing you genetic ancestry distribution, just to show you the diversity of the data in terms of genetic ancestry. And you have, we have individuals from a Colombian study. You can see that as a mark point, the European ancestry is the one in blue, the indigenous American ancestry is the one in red. Then you have the African in green and the East Asian ancestry in purple. And so you can see how just follow one of the colors, especially the indigenous American or European, you can see it's all over the place depending where the study took place. And the total of individuals we have so far in these studies is 18,000 people, give or take. And so the graph on the left includes the polygenic risk scores based on published, the Madhavat paper polymorphisms that overlap with the data we had from Hispanic Latinx. And the one on the left doesn't include the polymorphisms that we discover in the Hispanic, Latina, Latinx GWAS, and the one on the right we added. These are 2 polymorphisms, only 2. And I put a bar in red where the 63% area under the curve would be. And you can see we have 3 different PRS, one with the largest number of SNPs in purple, and then the green one is the one with the more strict p-value cutoff for inclusion into the PRS, which in this case includes about 164 polymorphisms. And so without including the 2 Hispanic Latinx specific SNPs, you can see that the area under the curve is mostly below the 62%. When we add the 2 polymorphisms, you can see that it moves closer to the red line. And you can see the top set of plots is for the US, then you have Peru, Mexico, and Colombia. And so you can see clearly that in the US and Colombia, it looks like it's moving more towards the right in the first graph. And then adding the 2 SNPs, everyone improves. A lot of improvement, though, you can see in the Peruvian sample. And this is because of the ancestry relation, the allele frequency differences for the different populations based on ancestry. The Peruvian is the one with the most indigenous American ancestry. But when we divide samples based on proportion of indigenous American ancestry, you can see that those with the highest proportion on top do the worst in terms of area under the curve, and it improves quite a lot when we add the 2 indigenous American polymorphisms. The other interesting thing to notice is those with the lowest indigenous American ancestry proportion are already doing better without the Hispanic Latinx SNPs, but they actually improve quite a lot as well when you add the 2 polymorphisms. So just to summarize this part of the presentation, the PRS in Hispanic Latina women is based on variants discovered in studies with other ancestries, can improve when you add population-specific variants. And based on this, the idea would be that as we are able to increase sample sizes for genome-wide association studies in Hispanic Latinx, we might find some more variants like this that could improve the PRS further. Clearly, for this, we need very large studies. If we think about the sample size, where when the European studies, I think the first GWAS was published in 2007. They found one variant in that GWAS. That was the paper, and now they have hundreds because they had those studies are based on more than 100,000 cases and 100,000 controls. The sample size we have for Hispanic Latinx populations are at the order of 10 times less, and therefore that hurts the ability to find these population-specific variants that clearly exist, as we demonstrated with this one we were able to find. Another important message I take from all this work is that for breast cancer, this doesn't mean it's the same for all traits, but for breast cancer, if the PRS based on European variants was already informative for Hispanic Latinas. When we add even 2 population-specific variants, it becomes equally accurate than in the case of Europeans. So we should be using this PRS. Some companies are already giving women their risk score based on these markers. We should definitely make sure that Hispanic Latinx women are also getting the results back because it's helpful for them. And the last point in my outline was I wanted to discuss the complexity of investigating difference and disparities by racial, ethnic category and genetic ancestry. And to kind of bring up this discussion or make my point about this complexity, I want to just briefly show you an analysis I did based on the observation. This is a long time ago. Data was based on, it's 2003 hazard ratios from a paper by Lee et al., where they were showing by racial ethnic category, again, based on census categories, that Hispanic Latinx individuals had a higher hazard ratio. And again, other groups also have a higher, even higher hazard ratio compared to non-Hispanic whites, but I was really interested in Hispanic Latinx. So based on the work I had been doing on risk, my approach was maybe I can look at the association between genetic ancestry and survival in Hispanic Latinas. We knew the social determinants explain a lot of these disparities in survival or mortality. That was already demonstrated. Access to care affects the how you do once you have a disease. But the question was, do genetics play a part? And the first thing I thought of doing is looking at, and I had done this before when looking at risk, is what variables that are not based on our genetics could be associated with ancestry as well as mortality. So if you look here, you see things like socioeconomic status, level of education, even fluency with English or exposure to alcohol. All these more social things actually are correlated with genetic ancestry proportion. This is based on data from US Hispanic Latinas. So the higher your SES, the lower your Indigenous American ancestry proportion, the higher your education, the lower your Indigenous American ancestry proportion. Hispanic Latina woman with higher Indigenous American ancestry drank less alcohol, and English used also went down with increasing Indigenous American ancestry. I analyzed data from a study of Latinas based on registry data plus population-based controls from the Bay Area, and we included in the model SEA's education and also tumor characteristics, and still found this relationship between increasing Indigenous American ancestry and higher hazard ratio. So at the time, we were very cautious. We said this might be related to some germline genetic component that increases or decreases survival in Hispanic Latina individuals. There could also be other factors that we're not taking into account that correlate with ancestry. So that kind of possibility stayed with me, and I thought, well, the very important factor is not only socioeconomic status, but also if you have or you don't have access to care and the quality of the care. And I talked to my colleague Larry Cushy, and I said, can we look at the Kaiser cohort of Hispanic Latinas, because they are all in Kaiser, so hopefully they are all receiving similar care and have the same access. And when we did that, we found no correlation at all between the risk of mortality and genetic ancestry. So this kind of takes me to what I think are very important challenges that we encounter and when doing research focus on racial ethnic categories or genetic ancestry, that I'm not sure everyone keeps in mind every time they design a study or they are thinking about diverse populations. But there was a really interesting point made in Scientific American, but some young investigators about post-treatment bias, that first we should keep in mind when we conduct analysis and run regressions that race ethnicity is anterior to SES education, occupation, location, insurance status, etc. This is also the case for genetic ancestry, meaning if we're kind of describing disparities, we don't adjust for all these other things, because belonging to a particular group or people seeing you as part of that group can lead to all these downstream effects, like you having a particular status in society, a level of access to education, access to certain occupations. So when you're thinking about identifying disparities, I think you should not adjust for these other downstream things. You just should model them depending on your questions, for example, as mediators. The other challenge is racial ethnicity labels are not stable, especially for Hispanic Latinx individuals. And so in the same paper, they show that 40% of Hispanic Latinos, only 40% had the same race ethnicity mark in the 2000 census compared to 2010. So people change in the way they want to be labeled, or the way they want to answer to the census or many other surveys that we conduct. And then, it's important to keep in mind that ethnicity is, it's a social construct, it's a multidimensional construct that includes genetic ancestry. So part of the way we use it, we think, we see it as associated with genetic ancestry, which is different than race ethnicity, but it is correlated. So when you're doing studies and you think, oh, I'm measuring genetic ancestry, so I must be measuring something related to the genetics or the germline, it's really important to consider that's not the case, like I show with the example of the survival analysis. In that case, ancestry was very similar to race ethnicity category. And particularly because increasing ancestry is also associated from one particular group, could be associated with a stronger perception from society that you belong to one category or the other. So with that, I would love to hear your thoughts and open up for discussion and hear your questions. And of course, all this work is part of a huge collaborative teams, including many other labs around the US and in Latin America, as well as people in my lab that do some of the analysis that you see.
Great. Thank you so much, Dr. Fejerman, for a really terrific talk encompassing a huge amount of work. We thankfully have some time for questions and discussion, so we'll open it up now. For those of you who feel comfortable, we certainly invite you to turn on your camera so we can see each other and have a little bit more interactive discussion. Just to remind again what we said at the outset, if you could type your question in the chat box or use the raise hand feature found under reactions and wait to be called on. And then when you're called on, if you could unmute yourself and ask your question, that would be wonderful. And we'll try to get to as many questions as we can in the time that we have remaining. Maybe I'll start just to get us started. So that was a really terrific talk. And I think you presented a lot of data about the ways in which polygenic risk scores and other classifiers perform quite differently across different population groups. I'm curious, I think a related idea is that I think different population groups often have a different level of willingness to undergo genetic testing and counseling. And then I think risk communication is in general quite hard, especially multifactorial risk. And I think to some degree is kind of culturally encoded across population groups. So I'm just wondering to what degree your team and your work has sort of engaged with those kind of ancillary aspects of the work that you presented this morning.
Yeah, I didn't want to talk about everything, and today I wanted mostly to make the point about race, ethnic category, and ancestry, because I think it's very important. but I've been working for the last 8 years on a program to educate Hispanic Latinas about hereditary breast cancer, and really started as a collaboration with my friend Isabel Duron, who came to me and said, Laura, precision medicine is all about genetics. Latinos are not gonna know, what to do with this information. We need to teach them about genetics so they are prepared. And then I decided to focus more on genetics of breast cancer, because I thought like going around teaching genetics more generally was unfocused. And we developed this program called Tu Historia Cuenta that is ongoing. We have reached about 1,800 Spanish-speaking Latinas in California, where we keep going. We're hoping to expand, and it's really fascinating to see how women are so hungry for knowledge and information, and ready to, we're testing high-risk individuals. We're actually being able to test them for those with strong family history, and it's going well. So I think it's been demonstrated that they are less likely to be tested, like about 4 times for Latinas compared to non-Hispanic whites. But if you bring the services, if you help them with access, if you bring the information, they are very happy to know what their risk is and go through the procedures. So that's kind of a long answer, but and there's other people working on this. And my colleague Christy Graves and Alejandra de Mendoza have programs also to train promotores about hereditary breast cancer, so they can spread the word in the Hispanic Latinx community.
Great, thanks so much. So we'll continue to wait for questions either from the chat or through raised hands. Maybe I'll ask another one just to sort of continue while we're waiting. I think so much of your work has so nicely realized these kind of thematic overlaps between women in Latin America and also Latin Americans living in the United States. And I had the sense from the work that you presented this morning that there's a lot of kind of thematic similarity in some of the findings between these different groups. I'm wondering if there are places where your findings really differ among Latin Americans living in the United States versus those who are resident in Latin American countries. I don't know if that was specifically elaborated in your talk, but would be interesting to hear about.
Yeah, so there's work done more based on like registry data showing that, for example, risk is higher in US-born Hispanic Latinas versus foreign-born Hispanic Latinas. My work in Latin America so far shows that, what the data shows, which is, globally, which is incidence is lower in countries with more indigenous American ancestry. But what I'm learning is different, and I think it's really important, is the distribution of stage at diagnosis in the US versus Latin America, for example. And in Latin America, the stage distribution is totally switched, kind of is shifting to the higher stages compared to what we see even for Latinas here. So here we're being able to still diagnose, they are diagnosed at a slightly higher stages, but it's not as dramatic as what we see in Latin America. And again, it has to do with access to screening. In Peru, for example, most of the diagnosed cancer is stage 2 or 3. There's a lot of stage 3, and it's because women who are in remote regions are just not being able to access. These countries have cancer control programs that are trying to address these issues, but again, infrastructure, funding, if that's a problem for us, imagine for low middle income countries.
Great, thank you. It looks like Maggie has a question. Maggie, why don't you unmute and ask your question?
Thank you, Satish. Thank you, Dr. Fejerman. It was a really nice talk. So I just had one follow-up question on the notes you were making before this last question on patient education. And I'm curious if you can speak, a little bit more about the provider piece of that and how you bringing the providers into the conversations that are being had with patients regarding the genetics of breast cancer in this case? Thank you.
Well, that's a wonderful question. We are adapting the Tu Historia Cuenta program to a clinic setting. So the first version of the program was based on educating community health educators, promotores, and having them reach out to their communities and just do the educational session. And then we did some surveys that the promotores apply, and we score the individuals. And then when we were trying to get them into genetic counseling and testing, it became a barrier. People either didn't have a usual source of care, they kind of called into clinics and they never heard back for appointments about genetic testing. So I was able to put together a pilot kind of just to solve the emergency that I felt we had, and I'm testing the people in the program on my own. And then, helping them if they have a mutation, the idea is we help them access the care they need. It's kind of a hand-holding, not a system solution. So because of this, I now implementing a new version with 2 clinics, one in San Diego and one in Sacramento, just as a way of testing, where we're adapting everything to the clinic setting. So we evaluated the clinic flow, we evaluated what they have in place, we evaluated their barriers, and then adapted everything to what they have. And we just finished the kind of the adaptation phase, and right now it's starting the actual implementation. And the idea is now that it's based on people who have a home, who have a community clinic where they go to. And we're also doing refreshers for the physicians because we survey them and they are not like up to date with all the information on what to do or how to think about family history and how that affects risk and what recommendations follow. So we're working on it. I can't say that I don't know if it's going to be successful or not, but we're trying to find a model that works.
Great, thank you. Patty, looks like you have a question.
I actually have a comment. Just Dr. Fejerman, I thought that was a lovely presentation, and I just wanted to send such good kudos to you, kind of pulling out some of the epidemiologic kind of methodology concerns in a lot of this research in the past, especially of thinking of race and ethnicity and genetic ancestry, not necessarily as confounding effects that need to be controlled for, but actually mediating effects, and to really frame the analysis according to the question you're asking appropriately. So I just wanted to say congratulations. I thought that was just a wonderful approach, and I think really meaningful now in terms of moving these fields forward. So thank you.
Thank you. I actually made it what I've noticed, because I'm an anthropologist and actually studied social anthropology and then became more of a geneticist. I feel like there's been a lot of conflict between social scientists and basic scientists about what genetic ancestry means, or race, ethnic categories, and how we talk about things. And I feel like there's people are not in disagreement. They're just are thinking differently and coming from different perspectives. And so I think that's what I was trying to do, is to show that it's question driven,
right?
Exactly. And what's important is to understand the constructs, whatever they are.
Exactly. And I think that's I think this, what you just said then to me is an extreme, like public service announcement for the value of having multidisciplinary and interdisciplinary approaches to understanding some of these complex problems. So yeah, I think this is great.
Great, thank you. There's a related, I think, question in the chat from Vidya Vedam, and she's asking about if you have insights about how research that describes genetic ancestry links to specific traits or diseases can be reported in a way that avoids certain groups being stigmatized or discriminated against. Do you have thoughts about that from your own work?
I have a lot of thoughts. I think we're all always evolving. So sometimes I look at papers I wrote, 15 years ago, and I think, oh, I could have put this in a better way. I could have written about this in a better way. And you see me, I use categories. Sometimes I say Hispanic Latinx, sometimes I say Latinas, Latinos, is because I think we all use many categories, and today I didn't do it, but usually what I said at the beginning of any talk is I say Hispanic Latinx sometimes because some people prefer that term, but other people from Hispanic Latinx background don't. And so especially older people like me, we were comfortable with saying Latina woman, but that might be exclusive for, might feel like I'm excluding some people when I talk. So I try to kind of change my wording and show inclusivity, as well as use some terms that part of the people would like to hear. And it's not, I feel like we have to kind of clarify these things every time we do a presentation before we even talk about genetic ancestry of what that means. And so I think that to avoid confusion, I think it's this part of introducing your terms or categories of what you're gonna mean or why you're using those terms. Why do you decide to look at genetic ancestry? What are you thinking? So if you present it in those terms very explicitly, and you say, well, there's germline variants that are not often seen in every background, and I'm very interested in finding those, and that's why I'm focusing on genetic ancestry, even though I know there's other factors that are correlated, and I write that clearly in my papers, then I think it makes sense. But still today, I'm reviewing articles from basic scientists who do not take the time to explain what they mean. And they say there's like Hispanic cells and European cells. And they confound ancestry with race ethnic category. And I think that's the problem. I think the problem is not in the words. The problem is in the use of the words and how we explain them.
Great, thank you. I'm also happy I'm not the only one who sometimes wants to rewrite papers that I wrote a while ago. So we probably have time for one or two more questions if there are any. Geraldina, I see your hand is up.
Very interesting. I don't know which term I like, Latina, Latinx, one of those. But actually my question was, maybe I misunderstood you, but when you presented your data showing the two SNPs that kind of helped in the indigenous population, my understanding was that it was looking at indigenous people from Peru and Mexico. Or did it include other South American indigenous groups, like from Brazil and so on?
Yeah, so when I say indigenous American, in this case, these variants are they came from some ancestral Asian populations during migration from, through the Bering Strait. So they are kind of shared within all the indigenous American backgrounds. So I had the opportunity of looking at this variant in, Argentina, Brazil, Uruguay, Chile. I didn't present the data because it's not published, but part of the consortium, we have this polymorphism for all the studies, and it's always present. This variant is in the root, every indigenous, even I think we saw it in a couple of individuals with indigenous ancestry from North America, so Native Americans. I don't know what frequency it is, but I think it's there. It's in that background too.
Okay, that makes sense. That wasn't.
And it's present in Asia, so in China it's present at about 1%. And there's some East Asian populations that have a higher percentage, as I saw this in the HEDP data from a colleague. So about 5% in certain groups. So it's an ancestral allele that came from Asia. into the Americas. It's just for some reason that we don't understand. It could be just random chance, which happens all the time in the genome. It became a lot more common in the Americas.
Quite interesting. What about us Latinx people that probably have little indigenous? Have you looked at, see if we have any ancestral type genes that?
No, so what happens is basically the European Latin Americans are similarly in risk as the Europeans. So if it's actually interesting, if you look at incidence data from places like Uruguay, Argentina, they have on average, Argentina is very diverse. So the north is similar to Peru in terms of indigenous American ancestry proportion, but the registry data comes mostly from certain cities with more European ancestry. And so incidence there is kind of equally high as Europeans, non-Hispanic whites in the US or Europeans. If you look at the map, you'll see that. So we don't have that protection if we have high European ancestry.
Okay, thank you.
Bummer.
It is.
Okay, I think not seeing other questions in the chat or hands raised, maybe we'll start to bring this to a close. I really want to thank Dr. Fejerman again for presenting all of this work, and to everyone for joining us this morning, I think for a really outstanding seminar. This is an opportunity just to remind you all to join us if you're able for our November seminar, which will feature a friend and colleague, Dr. Anna Xu, who will talk about her work to improve diagnosis of Burkitt lymphoma in East Africa. Again, and then our December seminars, Dr. Christian Ntizimira will be talking about some of his work related to palliative care in the Rwandan context. Once again, please do visit our website, cancer.gov/global-health, to find information about upcoming sessions, past recorded sessions, funding opportunities, events, news, and more. And follow us on Twitter, X, whatever it's called now, at NCI Global Health, and also on LinkedIn. So we'll end there. Thank you again, Dr. Fejerman, for joining us this morning. And thanks everyone for joining. Hope to see you at our next seminar in November.
Thank you for having me and listening to me.
Our pleasure. Bye, everyone.